Immunity is the capability of organism to resist harmful microorganisms from entering and damaging it. It also destroys old, non-functioning cells. We differentiate two types of immunity, cell-mediated and humoral immunity. These two types create innate and adaptive immunity. Innate immunity is represented by macrophages, neutrophils, monocytes, dendritic cells, natural killer cells, and a complement system. That's basically a system of plasma proteins which are working as targets for immune cells and are also creating complexes that are helpful in killing some bacteria. Adaptive immunity is formed by T cells and B cells that produce immunoglobulins, also called antibodies. In this video, we are going to focus on T cells. Thymus is the organ located behind the sternum, and it's part of the lymphatic system. We're going to hear more about this system in next video. At this moment, remember that the lymphatic system is important in providing adaptive part of immunity. Thymus is central organ, where all T lymphocytes mature. They incorporate proteins into their membranes, and these proteins work as receptors, which are very useful in transferring signals between cells. Thymus has its own capsule on the surface, which is made of dense, irregular connective tissue. Septate that extends from this capsule divide thymus into lobules. These lobules aren't separated. They communicate with each other, so that's why they are sometimes called pseudolobules. Reticular epithelium creates supporting structure. These cells, reticular epithelial cells, have got really light nucleus and well-seen nucleolus. Their long processes are connected, and they play an important role in differentiation and maturation of T lymphocytes. Each pseudolobule can be divided into cortex and medulla. Medulla contains something called Histol bodies, which basically are concentrically arranged reticular epithelial cells. These cells degenerate in the inner layer and show signs of keratinization and dystrophic calcification. That is accumulation of calcium into pathologically changed tissues. But not every circle in medulla is Histol body. You can also find vessels there. We've already mentioned that if the T cell wants to become mature T cell, it has to synthesize several receptors on its surface. T lymphocytes come from bone marrow, where all immune cells are made. They go to the thymus and create receptors. The major one is TCR, or T cell receptor, then CD3, CD4, CD8. CD means cluster of differentiation. We can differentiate T lymphocytes based on which receptor do they have. CDs work as co-receptors. They're helping T cells transferring the signal. T lymphocytes that activate CD4 is called helper T cell, and the one that activates CD8, cytotoxic T cell. In the beginning of the journey, T cells produce both receptors. They really just don't know who they want to be. Both receptors are inactive, so that's why this lymphocyte is called double negative. When the T cell comes in contact with the cell which produces MHC class I receptor, it will become cytotoxic T cell. MHC1 means major histocompatibility complex, and this receptor is located on every nucleated cell in human body. There are some exceptions from that. Cells have to synthesize and degrade proteins for most of their time. They degrade them via proteasomal digestion, and their parts, also known as antigens, present on the MHC class I. Antigen in general means substance that can generate immune response. Immune cells control these receptors, and when there's antigen that is somehow damaged or contains foreign substances, they destroy the cell. So, cytotoxic T lymphocytes are controlling MHC class I. T lymphocytes can also meet in thymus with cells with MHC class II receptor, and when they do, they become helper T cell. MHC class II is specific for cells called antigen-presenting cells, APC. One of the most known APCs are dendritic cells. These cells can eat different substances or whole bacteria, degrade them via lysosomal digestion, and part of them present on MHC class II. The role of T helper cells is to communicate with these antigen-presenting cells and send messages to other immune cells. Only some antigens are able to get in contact with the lymphocytes because there is a strong barrier between blood and cortex in the thymus. Cortex contains only capillaries with continual layer of endothelial cells and strong wall. You cannot find this barrier in medulla. Venules with high endothelium are located between cortex and medulla. Mature T lymphocytes are using them as a passage to the blood circulation, where they can reach thymus-dependent organ on the periphery. And you can see the hustle body once again. "99% of the lymphocytes die in the thymus. When T cell is unable to bind antigen, it undergoes apoptosis. That's controlled death of the cell. This is called positive selection, and it happens in the cortex. T cell that is binding more than one antigen or is binding to self-antigens too tightly dies. This is negative selection in medulla. This process is strictly regulated, and only perfect T cell can cross to the circulation. This sounds kind of rude, but you don't want to have T cells in your body that are hyperreactive or don't react at all. Am I right? Thymus is the organ that undergoes atrophy. It shrinks, but never disappears. Thanks for watching, hope you enjoyed the video. If you want some more, please let me know. If you liked the video, please subscribe. I also created a fan page on Facebook called Felschestein, where I'm uploading new videos. Bye!
Bye!